This is a great, great pleasure for me to introduce Professor Sir Mark Pepys, the first knight I am introducing ever, beside all the figure legends I usually introduce to my three sons. So it is really an honor for us to have you here today. Mark Pepys was educated at Trinity College in Cambridge, where he was a senior scholar and took a double first in the natural sciences tripos. After qualifying in medicine at the University College Hospital Medical School, he returned to Cambridge as a research scholar at Trinity for his PhD in immunology. He was elected to a fellowship in Trinity in 1973 for his discovery of the role of complement induction of antibody production. After training in clinical medicine and continued immunology research at the Royal Postgraduate Medical School, he was appointed head of the Department of Immunology at the Royal Free Hospital School of Medicine. In 1977, he returned to the Royal Postgraduate Medical School as senior lecturer and established the Immunological Medicine Unit, which became one of the world's leading centers for basic and clinical research on the acute phase response and amyloidosis. In 1999, he was appointed professor and head of the Department of Medicine at the Royal Free Campus of UCL and moved his whole department there to establish the UCL Center for Amyloidosis and Acute Phase Proteins. He also created the UK NHS National Amyloidosis Center, funded by the UK Department of Health to provide diagnostic and management advisory services to the whole national caseload, and which also sees many patients from abroad. He received generous support from the MRC and the Walpole Foundation and the Wellcome Trust, including one of the first seeding drug discovery awards. He is a fellow of the Royal Society, a founder fellow of the Academy of Medical Sciences, and has been a member of the councils of both academies. He is the winner of the Royal Society GlaxoSmithKline Prize in 2007, received the 2008 Ernest Chain Prize for Medical Discovery. He is the director of the new UCL Wolfson Drug Discovery Unit and was created Knight Bachelor for Services to Biomedicine in the 2012 New Year Honors and was elected to an honorary fellowship of Trinity College, Cambridge in 2014. Thank you very much for coming, and we look forward to hearing you.
Thank you very much, Ronit, for that excessive introduction. I'm exhausted just listening to it. I'm sure the audience are probably half asleep by now. It's a great honor and a pleasure. Thank you, Udi, and the rest of the team for inviting me to talk at this meeting. I've enjoyed the meeting so far. A lot of very interesting talks and very good science, really fascinating. Before I start, I want to apologize for one thing, and that is wearing a tie. I noticed that I'm obviously the only person here wearing a tie. I actually am very familiar with Israeli society, and I knew that this was an extreme thing to do, but it's my birthday on Monday, and a colleague of mine, thank you, a colleague and friend of mine for the last 50 years or so, he sent me this tie, and it's got on it some capsules and syringes and a stethoscope all over the tie, and I like it because my memory's not what it was. And when I sometimes forget what I'm supposed to be doing, I look down at my tie, and it reminds me. So if you see me looking down here, you know, it's for that reason. Let me get my talk. Hang on a second. Right, so I'm going to talk about academic drug discovery, how to get beyond academic drug discovery. And the first thing to say about it is that it's the most challenging, most difficult, most frustrating activity undertaken by the human race. So if you're not really, really determined to do drug discovery, you need to change your job and do something else. It takes forever, and it costs a fortune, and nearly always fails. The only thing that keeps one doing it is because it's a really good thing to do. If you can make a new medicine for unmet medical need, that's a great achievement. But it does require extraordinary determination and persistence, and serendipity, which we heard about from Professor Noodleman very eloquently this morning, and I'm going to say some more about that in just a second. But what I've made on this slide is a sort of list of the things with which everybody's familiar, but from the point of view of academic contribution to drug discovery, what we do is the first bit. I mean, the understanding of the pathobiology of disease. Sorry, let me go back. The pathobiology of disease. That happens in academia and in research institutions. It doesn't really happen to any significant degree in big pharma or even in small biotechs. The ideas and the new discoveries, the people who write the papers in Nature and Science and so on, they're in academia. And there's been a tremendous evolution since the time 30 years ago when I started trying to do drug discovery in the relationship between academia and industry, which is some of what I'm going to talk about. So serendipity, this is a great word. It was invented by Horace Walpole on the 28th of January in 1745. It came from this fairy story, The Three Princes of Serendip, which you've already heard of. Serendip is Ceylon, Sri Lanka, as it's now known. That's where this supposedly happened. And the word means the making of discoveries by accident and sagacity of things not sought. So accidental, discovery, good luck. And various famous people have described it in different ways. Louis Pasteur said, in the field of observation, chance favors only the prepared mind. You have to be prepared to see that the accidental finding actually means something and not ignore it. Asimov, the science fiction writer, said, the most exciting phrase to hear in science, the one that heralds new discoveries, is not eureka, but that's funny. And the best one is Julius Comro. Serendipity is looking in a haystack for a needle and discovering a farmer's daughter. You probably shouldn't say that nowadays. It's very sexist, so I apologize to anybody who's offended by that one. But he wrote it quite a few years ago. So I have the good fortune that the targets that I have been trying to make drugs against are plasma proteins. These are wonderfully accessible. They're in the circulation. It's the easiest place to get a drug to go. You can squirt it straight into a vein, and there it is. And that makes life a lot simpler in many ways. And there are two proteins that belong to the pentraxin family, serum amyloid P component, SAP for short, and C-reactive protein, CRP for short, that I have been working on for the last 40 years. And another protein, which is transthyretin, a normal plasma protein that everybody has, which actually happens to make amyloid, because it inherently has a propensity to do that, and I'll say some more about that. So those are the three drug targets that I have been trying to hit for many years. I'll start with amyloidosis. So systemic amyloidosis is a relatively rare but usually fatal disease. It kills about 1 in 1,000 people who die in developed countries. So it's rare, but it's not vanishingly rare, and it's a major unmet medical need because a substantial proportion of the patients still die within 6 months of being diagnosed. 25% of the patients die within 6 months of being diagnosed. A really bad disease. And it's caused by circulating plasma proteins, normal soluble globulous proteins, which misfold partly and then aggregate into insoluble abnormal conformation that's called amyloid fibrils, which have a cross-beta sheet structure and which are relatively resistant to degradation. And it can be produced by both normal proteins and abnormal proteins. So you can either have prolonged normal exposure to a particular protein, transthyretin, that we all have, and that eventually makes amyloid in nearly everybody if you live for long enough. Or it can be overproduction of a normal protein, and there's a particular protein called serum amyloid A, which is an acute phase protein, and if you have chronic inflammation for many years and produce an excess of that protein, it will eventually turn into amyloid in a certain proportion of people. And then there are abnormal proteins, which can be either an acquired abnormality as a result of a somatic mutation, making an abnormal protein, or you can have inherited genetic variants of proteins, which because of their variant structure, have a propensity to misfold and aggregate as amyloid fibrils. And these fibrils accumulate in the tissues, and mysteriously they are very poorly cleared, if at all, by the body. This is a stark contrast to the body's normally very efficient mechanism for clearing debris from the tissues. If you damage yourself, have a bruise or break a bone, and you have a lot of dead cells and blood in the wrong place outside in the extracellular space, provided it doesn't get infected, all this debris gets silently cleared away by macrophages which come and clear it up. Very marvelous mechanism. It just does not happen with amyloid deposits. And so the natural history is that the amyloid deposits accumulate, they get bigger and bigger, more and more, they disrupt the architecture of the tissues and eventually the function of the tissues, and you get disease. Now, in the 1980s, when I had been studying amyloid for maybe half a dozen years, I had an idea, or maybe 10 years, I had an idea for a novel therapeutic approach to it. And I hawked this idea around all the big pharma companies. I said, would you like to work on a new treatment for amyloidosis? And they said, what is amyloidosis? They didn't know what it was. They weren't interested in it. They weren't interested in developing any drug that would have a market value of less than 300 million pounds or so in the 1980s. Now, if you that would be, say, 3 or 4 times as much nowadays. In the 1990s, matters had evolved so that they were open to ideas from academic people, and I had some fruitful collaborations with Pfizer. We had a drug called Miridesap, which was an anti-inflammatory drug, potentially of great use in rheumatoid arthritis and other inflammatory diseases. When they had spent $500 million on it, it had a side effect, which was renal tubular proteinuria, which was no big problem, and as soon as you stopped the drug, it went away, and it only occurred in a few people. But the FDA didn't like it, so from one day to the next, they stopped the project. Threw away $500 million of investment and a very useful drug. I worked on them with an inhibitor of CRP. I'll say a bit more about that later, for acute myocardial infarction and other things, and that didn't go anywhere. We did a high-throughput screen, which I had invented, didn't find any hits, and they stopped that. In the 1990s, I collaborated with Roche, and there we had a wonderfully successful collaboration, and within a short space of time, we had a potential drug, which is still going on, and I'll tell you some more about that. They wanted it for Alzheimer's disease. I wanted it for Alzheimer's disease, but as you've heard, that's a very challenging disease. And I also wanted it for amyloidosis, which is also very challenging, but more realistic potentially for therapy. And then in the 2000s, with the blockbuster famine, and poor pipelines, and greater awareness of rare diseases, pharma became quite interested, in fact enthusiastic, about academic ideas. Pfizer gave Stanford and MIT hundreds of millions of dollars in exchange for first right of refusal to all the new IP that came out of their centers. AstraZeneca moved their whole headquarters to Cambridge universities to be close to the epicenter, the laboratory of molecular biology, full of Nobel laureates and so on. GSK had a different strategy. Patrick Vallance, my former junior colleague, who became from one day to the next, one day he was my junior colleague, the next day he was the head of R&D at GSK with an annual budget of £1 billion and 3,000 people working for him and so on. So he had a completely different approach. He wanted to cherry pick individual scientists, leaders in their particular field, get them to collaborate with GSK. No money changes hands till you've got a successful drug, but you have an intensely collaborative, synergistic relationship, and this was a great idea, and surprisingly enough, I was the first person to be involved in this collaboration with GSK, and that's been going on since February 2009. I keep giving you these dates just to give you the feeling for how absolutely horribly long it takes to get anywhere in this business. So here is amyloidosis. It's a nasty disease. It progresses inexorably. The body can't get rid of it, and eventually the patients die of organ failure of one sort or another. And you can see why it's bad for you. This is a cross-section of a heart of a patient who died with cardiac amyloidosis. It's about 3 times the normal weight. It's extremely stiff. The heart has to dilate in diastole in order to fill with blood, in order to contract and pump the blood around the system. If it can't dilate to fill with blood, it can't pump so much blood, and so you have low output heart failure. This picture here is the heart of a patient of mine, explanted in order to transplant her with a new heart. She had end-stage heart and kidney failure. She had heart and a kidney transplant done at the same time from the same donor. That heart is grossly abnormal. It's sitting there like a plastic model of a heart. Normally the explanted heart would just collapse, a normal heart. This is sitting there solid, rigid, full of amyloid. And after this successful operation, she lived for another 32 years and got to see her granddaughter growing up and so on. So that was very successful, but it shows you the horrible things that amyloid can do. And what do we do about treating these patients apart from transplanting their organs? Well, we try and keep them alive and do everything we can to keep them alive long enough to try and get rid of the protein that is the precursor of the amyloid substance. And sometimes you can do that, and sometimes you can't. And the treatments to try and do it in certain types of amyloid are extremely toxic, and they sometimes kill the patients who are already sick just through the toxicity of the medicine. So this is extremely difficult. There are new approaches coming along trying to get rid of precursors by genetic interventions or stabilizing with small molecules the precursor proteins so that they don't misfold. And these things are being tried, but they haven't worked completely or at all yet. So removing amyloid is clearly a desirable thing to do. If we could get rid of these abnormal deposits, this would restore organ structure, organ function, and would be good for the patient clinically. And that's what I've been trying to do for quite a few years. And my interest in this is focused on that serum amyloid P component, SAP, that I mentioned earlier. It's a normal plasma protein. Everybody's got it. Nobody's ever been found who doesn't have it. There are no variants at the protein or the glycan level that have been described. And we've shown over many years that it contributes to both the formation and the persistence of amyloid. And therefore, it's a valid therapeutic target. We've solved its crystal structure. We know a great deal about it. The first thing I did with this protein was to discover why it was always present in amyloid deposits. So amyloid deposits have got the protein fibrils I've told you about. They also always contain SAP. And why is it there? That was discovered in 1965, before I was in the field. But in the 70s, I discovered the reason why SAP is always in amyloid deposits, and it's because it's a ligand-binding protein, and it binds in the presence of calcium specifically to all different types of amyloid fibrils. They obviously all share a particular structure, regardless of which protein they come from, and SAP recognizes that structure, and it binds to them. So diagnosing amyloid is a difficult thing. You have to think of it. And until we came along with this diagnostic test that I'm about to show you, you had to have a tissue biopsy. You have to take a piece of tissue, sometimes, usually, in fact, in those days, used to come after the patient had died. Nobody knew what the patient died of. And at autopsy, they looked under the microscope and they saw amyloid. If you think of it while the patient's alive, you have to take a piece of tissue, you have to do the proper stain and know what you're doing, and then you recognize amyloid. This is very unsatisfactory. When you take a big biopsy from somebody's liver, you're looking at 1/10,000,000th of the liver, let alone what's happening in the kidney, the adrenal glands, the peripheral nerves, anywhere else. So I invented a whole body imaging technique involving radio-labeling normal human SAP, injecting it intravenously into the patients, and image them in the gamma camera. And what you see here are a series of gamma camera images of patients who've had this test. This one here, the top left, is a patient with what's called AL amyloid. It's in the liver and in the spleen, massively enlarged, and it's throughout the bone marrow of this patient. Here's a patient with a different type of amyloid looked at from the back, and the amyloid is in the spleen and the kidneys, and this little thing here just on top of the kidney is the adrenal gland. You absolutely have to have your adrenal gland working in order to stay alive, and amyloid destroys it. This is a very dangerous thing to have, which you would never know about if you couldn't have this test to look at it. It's about the size of your fingernail, that gland. And here, very importantly, are two patients who clinically were identical, and they were suitable for a particular type of treatment which had at that time a 15 to 20% procedure mortality rate, so a fifth of the patients died from the treatment. And they had, although they were clinically indistinguishable, the one had amyloid just in the kidneys, the other had amyloid in the liver and in the spleen. This patient would have died from the treatment. That patient would have been a good candidate to have it. So this was a very important contribution to understanding this disease. And the most important single thing it showed us is that amyloid can actually go away. If you get rid of the precursor protein, the amyloid can go away. So here are two patients who were successfully treated to get rid of the precursor protein, and the amyloid in the liver, you can see over a couple of years, has largely gone away and much reduced in the spleen, and another one with the same thing. So that's quite encouraging. My idea for treating amyloid came from the observation we made when we discovered for the first time a defined chemical ligand that could inhibit the binding of SAP to amyloid fibrils. It's a ligand binding interaction. I looked for and found a small molecule, it's cyclic pyruvate acetal of galactose, which specifically released the binding of SAP from amyloid in vitro. Obviously, this couldn't be a drug. There's nothing druggy about this little sugar molecule. But the idea was there to dissociate the fibrillar and non-fibrillar components of amyloid. And eventually, in collaboration with Roche, we made such a compound designed to do that. And it was reported in Nature in 2002. And this is the compound. It's bis-D-proline hexanoyl bis-D-proline. So two D-proline molecules joined by a hexanoyl chain. It's a palindromic molecule, and I gave it this palindromic acronym, CPHPC, for carboxypyrrolidine, hexanoyl pyrrolidine carboxylate. And it is bound by SAP with a very good KD. And it's now on its way to being a drug, and it's got its WHO international non-proprietary name, Miridesap, there. And we did the crystal structure, the complex between the drug and SAP. This is 2 pentameric SAP molecules joined by 5 of these palindromic drug molecules in a very high avidity complex. And something very extraordinary happened when we first put it into patients. As soon as you expose patients to this drug, the plasma SAP concentration falls dramatically and virtually disappears completely. And if you keep the patients on the drug, as long as you do that, and we've had patients on it for up to 7 years, SAP remains essentially absent from the circulation. This drug did something unique, unprecedented, and unfortunately not repeated. I wrote a patent in which I expanded the claims to this being a general method for getting rid of an unwanted protein from the plasma. Cross-link these plasma protein molecules with bivalent ligands, and you'll get rid of any protein. Unfortunately, not true in our experience so far. It works brilliantly for SAP, but not so far for anything else. And for SAP, the reason it works is that the complex of 2 SAP molecules in that complex with the drug is recognized as abnormal by the liver cells, and they take up the complex and destroy it completely. So this is extremely efficient. It was the American Chemical Society Medicinal Chemistry Highlight of the year. It's a new pharmacological mechanism, and the question is, does it do any good? Well, it certainly doesn't do any harm. First thing for a drug is it shouldn't be bad for you. And this drug has had no adverse clinical effects in over 100 patient years of experience now, and nor has the depletion of SAP. Nothing bad happens. And plasma SAP is depleted throughout the treatment, but the very avid binding of SAP to solid phase amyloid and the very fast PK of this drug combine to make it impossible for the drug to remove all the SAP from the amyloid deposits. So we have not yet tested my hypothesis, which was that if you removed SAP from amyloid, the body would get rid of the amyloid fibrils. Haven't tested it, and so far it hasn't worked. So the patients suffered no harm, but no amyloid regression occurred. And I therefore went on over the years to think of another approach to eliminating amyloid deposits. And in 2005, I invented it. The idea is that the drug removes circulating SAP, but it leaves some SAP in the amyloid deposits. And you could then safely give anti-SAP antibodies to the patient, and they would target the residual SAP in the amyloid deposits. Here we're dealing with SAP just as a passive antigen target, just as a specific marker of amyloid deposits. It's got nothing to do with any pathogenic possible role of SAP. It's just a target. The antibody binding, the antibody I designed for this, was designed to be an antibody that would trigger complement activation, engage the macrophages, and tell them, eat me. And that's what they do. When I told my team in the lab meeting, we're going to do this experiment, human SAP transgenic mice, we'll give them amyloid, we'll give them CPHPC, we'll deplete the SAP from the blood, we'll give them a big shot of anti-SAP antibody, one of 2 things can happen. Either the mice will explode, because we've produced massive inflammation wherever there's amyloid, or else they'll get better. The macrophages will do what they're supposed to do and eat up the amyloid, and that's what happens. So here we are. This green stuff is the amyloid stained specifically, replacing almost the whole spleen in a mouse in this experiment. And 28 days after a dose of control IgG with no antibody activity, the amyloid is all there. 28 days after a single dose of anti-SAP antibody, the amyloid is all gone. There may be a tiny speck here and there, but basically the amyloid has cleared away, and nothing bad happened to the mice. None of them get sick, none of them die. And after being patented, et cetera, and licensed to GSK for development, this paper came out in Nature reporting the finding. And this is the mechanism by which this treatment works. Here are a series of images of toluidine blue-stained sections of liver from the mice in these type of experiments. This first one is a mouse that's had no treatment at all, so before the treatment. The pink stuff, amorphous pink blobs, is the amyloid in the liver. And you notice that there are no inflammatory cells showing any interest in it whatsoever. That's why it doesn't clear away. The cells that should clear it just ignore it. 1 day after the dosing with antibody, every single bit of amyloid is completely surrounded by mononuclear inflammatory cells, macrophages, all around them. Within 2 to 3 days, and here by day 5, most of these macrophages have fused into multinucleated giant cells, which are engulfing and surrounding and degrading the amyloid. 2 weeks later, it's all gone. No amyloid, no cells, normal histology restored. It's miraculous. So February 2009, the patents were licensed to GSK. They fully humanized the anti-SAP antibody. They did all the necessary workup, phase 1 with the CPHPC alone and followed by the antibody, and the results in humans are exactly like the results in mice. It makes the amyloid go away. This was reported in, first of all, in the New England Journal a couple of years ago. We've got the full write-up of the phase 1 study about to be accepted, I hope, for publication in another high-impact journal as we speak. And here is just one example of this. Here is a patient with a particularly bad type of amyloidosis called AL amyloidosis. Here is the scan. You can see her liver is absolutely solid with amyloid. And she was given a reasonably sized dose of the antibody, and 42 days later the amyloid is essentially gone from the liver. And you can see that the amount of tracer SAP localized there is dramatically reduced. That accounts for the different appearance. The extracellular volume, this extracellular space is where the amyloid accumulates, that's greatly expanded in amyloid, and that is reduced to normal, from above normal to normal, as you can see here. And the liver stiffness, greatly increased by amyloid, also is reduced. Every patient who's had an effective dose of this antibody shows a massive consumption of complement. Many grams of C3, the main component of complement, have been consumed, showing that the mechanism in patients is the same as the mechanism we showed in the mice. And they also get an acute phase response. These are acute phase proteins which go up when there's inflammation of any significant type anywhere in the body. So the mechanism in humans looks exactly the same as the mechanism in mice. Importantly, I'm not bothering to show it to you here, lots of little numbers, liver function improves too. So not only have we got rid of the amyloid, we've actually proved that what I told you is the pathogenetic mechanism of amyloidosis is actually, what I've told you is the right answer. There's some controversy. People talk about toxicity of amyloid and this, that, and the other. In systemic amyloidosis, all the evidence is that it's just the accumulation of amyloid, this passive stuff which expands the extracellular space, destroys the structure, and therefore the function. That's the pathogenesis. And we've proved it here. We got rid of that stuff, and liver function gets better. At the moment, there's a phase 2 clinical trial in progress focusing on cardiac amyloidosis, because that's the main thing that kills patients with these worst types of amyloid. And if it shows that it works there, then we'll be very encouraged, and it should go all the way to becoming a medicine, provided nothing bad happens. And just in case anybody's feeling too happy about what I've been telling you so far, if you haven't followed what's been going on with GSK, a couple of weeks ago, their new female CEO, I don't say that for any significant reason except that she is female, which is quite unusual for a major pharmaceutical company. She just stopped 30 major projects which have had millions spent on them and closed down the rare disease unit in GSK. And I was, of course, extremely alarmed when I saw the press release, and I sent an immediate email to my colleague Patrick Valance, who's the head of R&D, and said, are we at risk? And he wrote back to me immediately, no, totally protected. So for whatever reason, our particular project, which could have got dumped, is totally protected, and it's steaming ahead on all fronts. But it just shows you that this is a very perilous tightrope, and you can drop off it at any moment. Anything could happen. Okay. Transthyretin, which I've mentioned before, is another type of amyloid, fibril protein. Everybody's got this protein. If you look at autopsies of people at the age of 80 and over, they've all got some microscopic bits of this type of amyloid in their body, usually clinically silent. Occasionally, patients in the past used to be diagnosed with heart failure due to cardiac amyloid made of transthyretin. It was thought to be incredibly rare. It turns out it isn't. It turns out to actually be quite a common disease, particularly in men. So men over the age of 70 presenting with heart failure with preserved ejection fraction, it's a particular type of clinical description of the heart failure. A high proportion of them have transthyretin cardiac amyloidosis, and we can diagnose that by a new sort of scan. This is a bone scanning tracer, DPD, and you can see that it actually localizes very specifically in the heart of people with this type of amyloidosis, and we can actually see by tomography the amyloid and the anatomy, pathology and anatomy all together, showing that in the ventricular wall of the heart. So that is a very important unmet medical need and a target for treatment. This is the crystal structure of transthyretin, and it's got 4 identical subunits, non-covalently associated, and it has 2 symmetrical identical binding pockets which carry thyroxine. That's called transthyretin because it carries thyroxine, thyroid hormone, and retinol binding protein, which carries vitamin A. And here are 2 thyroxine molecules inside the 2 binding pockets. Other people had the idea that you could stabilize transthyretin and stop it turning into amyloid by occupying those binding pockets with a molecule that fitted in nicely like thyroxine. And that has gone all the way to make a drug that's called tafamidis, and which is now sold by Pfizer, and it costs about 100,000 euros plus per year. It was licensed by the European Union. It was not licensed by the FDA because it doesn't really do much good. The evidence of efficacy was so limited that the FDA did not license it. But it's licensed in Europe, not used in UK because NICE, which is the body that determines whether the National Health Service will pay for a new medicine, said the risk-benefit, cost-benefit is not good enough, so it's not used in the UK, but other countries are using it. It's a good idea, but it doesn't really work so far. I was pursuing my idea that if we cross-linked pairs of transthyretin molecules, they would be cleared rapidly like SAP. That would be a wonderful treatment. Get rid of the precursor. You don't need transthyretin. And you would stop the disease just like that. So we designed some molecules. This is thyroxine. This is tafamidis, the drug. And we designed molecules which had something that would fit in nicely into the binding pocket in a typical palindromic structure like our CPHPC molecule. But serendipitously, the crystal structure on which we base this design, on which I based this design, I drew this on a piece of paper, came from a colleague, the guy who had invented tafamidis, and he'd got his crystal structure wrong. He had orientated this ligand alone in the binding pocket the other way round, okay? He had got it with the carboxylate and the dichloro rings in exactly 180 degrees the wrong way around. If he'd got it right, I would have made these head groups the other way around. But fortunately, he got it wrong, so I'd get my thing the wrong way around. And it didn't cross-link TTR at all, transthyretin at all. What it did is it went straight down the middle, so that we occupy both binding sites and the central channel between them. And this turns out to be not just a stabilizer, but a super stabilizer. This interaction is so avid that it's irreversible under physiological conditions. I mean, chemically, you've got to call it a pseudo-irreversible interaction, but there is no physiological condition in which you can dissociate it. It absolutely locks it down like a piece of stone and is magnificently better at doing what it was not intended to do. It's magnificently better at stabilizing TTR. It doesn't get rid of TTR, but it stabilizes it. And we wrote this up in the PNAS a few years ago, and of course we patented the hell out of this idea and so on. But it turned out that the initial molecule, not surprisingly, is not druggy. It's too big, it's too hydrophobic, and so on and so forth. And we worked with GSK quite a lot, made 600 different compounds, and got very close to having something which could have been a candidate for development, but then it had some cardiovascular toxicity liability, and that program stopped. Meanwhile, Vittorio Bellotti, who is a long-standing colleague of mine and works in my unit now with his own team, he has discovered actually what the mechanism is of transthyretin fibrillogenesis, and it's more complicated than everybody used to think. It involves a single proteolytic cleavage in one of the protomers, and then together with mechanical forces, the sort of thing you might expect to have in the tissues of the heart that's beating all the time, or the carpal tunnel in the wrist, which is another place that's moved around all the time in the human beings. That's where this type of amyloid forms. The combination of the cleavage and the mechanical forces, that's what produces transthyretin amyloid. And our palindromic or bivalent ligands that go down the central channel, they block it much better than anything else. So the single ligands can't really do it at all. You have to go down the central channel, which our molecules uniquely do. And here you can see in this little video that extraordinary little channel which they managed to thread their way down. It's a very clever trick. We don't really understand how they do it, but they do it very reproducibly. And the reason why our molecules are so much better is that the third binding site, these two binding sites everybody knew about, the third one we discovered down the middle here, the interactions down there produce amazing long-distance stabilization effects, which you can show by NMR, including the loop on the outside, which is where this critical proteolytic cleavage takes place. So there's quite a lot of understanding now in our group of the mechanism to prevent TTR amyloid from happening, and we're hoping to re-engage with GSK or get other investment, which is currently on offer to us, to develop these as a treatment for this type of amyloidosis. So moving swiftly on to another therapeutic program in our team. This is Alzheimer's disease. So we all know how difficult Alzheimer's is. The main thing to say about Alzheimer's disease is nobody knows what causes the neurodegeneration. You saw a list of some of the possible mechanisms, but nobody actually knows. We know people get demented because they've lost cerebral neurons, the neurons have died. We don't know why. There are various hypotheses. There's certainly a very tight association with A-beta amyloid because of mutations in the genes which produce more A-beta amyloid give you early onset familial autosomal dominant Alzheimer's disease. So that's very, very close to it. But is it the amyloid deposits? Is it prefibrillar aggregates which are neurotoxic? Nobody really knows. From my point of view, SAP is present on all these amyloid deposits, and not only on the amyloid, but also on the neurofibrillary tangles, which are hyperphosphorylated tau protein. These are the pathological hallmarks of Alzheimer's. They've all got SAP on them. Okay? We've got a drug that removes SAP from the brain and from the amyloid deposits, as it turns out, which I will show you. And that would seem to be a good thing to do, not only because it might affect the amyloid deposition, but also other people have shown, and we have confirmed, SAP itself is directly neurotoxic for some cerebral neurons. The amount of SAP in the brain is 1,000th of the amount in the plasma. So much less than you would expect for a molecule of its size. So it's surprisingly excluded from the brain, consistent possibly with it being neurotoxic, good thing not to have it in the brain, and everything that gives you dementia is associated with too much SAP in the brain. Living too long with a normal amount, being old, okay? Alzheimer's disease and other conditions where you've got amyloid in the brain, too much SAP, bangs on the head, American footballers, boxers, blood getting into the brain, strokes, all these are things which give you a higher risk of dementia. So SAP, probably bad for the brain. We've got a drug that removes it. Here is a section of normal age brain stained with anti-SAP, and you can see staining of SAP inside the body of apparently normal pyramidal neurons here, and bits and pieces that are bits sitting around little capillaries, which is presumably a post-mortem leak, but actually inside the neurons in a normal brain there is SAP there. So for many years I've been trying to get money to run a trial based on these ideas. 15 years, I finally got the National Institute for Health Research to cough up the few £1 million it needs to do a clinical trial. And this trial, called DESPIAD, Depletion of Serum Amyloid P Component in Alzheimer's Disease, is starting this month. 100 people in a double-blind placebo-controlled trial, Alzheimer's disease. Half are going to get the drug and SAP depletion, half will get placebo. And this is the basis for it. We did in 2005. 5 patients with Alzheimer's, we gave them CPHPC daily. SAP disappears from the blood, as it does in everybody. They were on it for 3 months, and we stopped the drug, and it came back. And here is the CSF, cerebrospinal fluid. Similarly, SAP was there before the treatment. It disappears completely, and it goes away. We can't look at what's happening to the amyloid, obviously, in the patients' brains, but in mice, where we've got models of Alzheimer's disease, we can do that. And what this slide shows is that SAP on the cerebral plaques of amyloid and in the cerebrovascular amyloid is all completely absent in the mice that have had their SAP, human transgenic SAP, depleted by our drug. So we know the drug will do biochemically what it wants to do, and now we're going to find out, we hope, in 3 years' time, whether it does any good to anything of everything that we're measuring in people with Alzheimer's disease. That says what I've just told you. So the final The final project I want to tell you about is C-reactive protein. C-reactive protein, which I started working on even before SAP in the very early 1970s, it's the classical acute phase protein. It means it's very low concentration if you're healthy and well. If you get sick for almost any reason, tissue damage, infection, injury, it doesn't matter what, the production of CRP increases dramatically and can go up by 10,000-fold in concentration in the plasma. It's very sensitive to what's going on, but has absolutely no specificity. Goes up in anything that's bad for you. It binds specifically to phosphocholine and to other ligands, but phosphocholine is particularly important because it's obviously the polar head group of the main phospholipids in this plasma membranes. And in cells that are damaged, phosphocholine is exposed in a way which it is not exposed in the membranes of healthy cells. So CRP binds to damaged cells. It activates complement, which is a major pro-inflammatory system in the plasma whenever it binds, and it could therefore exacerbate existing tissue injury. People speculated about that for quite a few years, but I showed it first. And here is the 1999 in this paper. If you tie the coronary artery in a rat, you give it a heart attack of a certain size. If you fill the rat up with human CRP before you tie the coronary artery, you get a bigger infarct. That was just a model system, not a very tractable one, I have to say. Terrible experiments to do properly, but convinced ourselves and others that that is what happens. And we know all about the binding of phosphocholine and so on. So based on the success with the SAP drug, I synthesized bisphosphocholene hexane. And that does for CRP exactly what the CPHBC does for SAP. Here is this original molecule. Here is the crystal structure of the complex, which is highly stabilized, and that is completely blocking the CRP from doing its ligand binding trick. And that works in the experimental model beautifully. So here's the heart attack size in the control rats. Here they are given human CRP, much bigger infarct. Here they are with CRP and the drug. It completely abrogates the enhancement of damage by CRP. And more rats survive, etc., etc. And this was on the cover of Nature in 2006. And drug development started. It proved to be impossible to synthesize and purify bisphosphocholene hexane at the scale needed even to do the preclinical toxicology. They're not very druggy-looking molecules. We actually found that bisphosphocholene octane was much more potent than bisphosphocholene hexane, which was really interesting, but that couldn't be done either. So the program languished for a while, and just lately we've come up with some really novel ideas, and we've got new palindromic molecules, nothing at all like phosphocholine, that are much more potent and are little white, beautiful white water-soluble powders and everything, looking much more like drugs. So we're optimistic now, and this chemistry has all been done by Wuxi very skillfully for us, and we're just about to ask them to make 20 or 30 or 50 grams of the best one so that we can do a proof of concept experiment where we do an experiment in animals where CRP makes injury worse, and we show that we abrogate it with the drug, and then we'll be ready to go back with my big hat in my hand to GSK and say, spend a few tens of millions of pounds making this into a medicine. So the story of academic drug discovery is that academic industry collaboration is essential. We are the ones who discover the new ideas and so on, but we haven't got the expertise and we haven't got the resources to do the development. It's not just the physical and financial resources to deal with the physical things that have to be done. It's dealing with the regulations, the environment, the regulatory environment in which drug development has to take place is a nightmare. It's getting worse all the time. It's not getting better. They make lots of talk about accelerated approval, blah, blah, blah. I don't see any significant evidence of it at all. So my personal experience over 30 years is that we've had some successes and some failures, different things. We haven't got any medicine yet after 33 years. You could say that's because I haven't been working hard enough, but I have been trying quite hard. It's very, very, very challenging. And the worst thing is that it could stop at any moment. More than 95% of drug discovery and development programs that start fail. They do not go all the way to a licensed medicine, and you can fail at absolutely any stage. There are various graphs about where you're more likely to fail, and if you get to a certain point, you might get a bit further down, but they can stop at any stage. All it needs is one patient to die or some horrible side effect to come along, or, you know, not the efficacy you thought you had, blah, blah, blah. They can fail, and they mostly do fail. So it's an absolute nightmare. Now, here we are, 2017. The scientific prospects are amazingly good. We can do extraordinary things. We can switch genes off. We can do terrifically clever things with antibodies and other biologicals, but there's no evidence to me that the regulatory and the commercial environment is getting any better. The final thing to say is, who's going to pay for these drugs? They're incredibly expensive to make, and they can't be made and sold cheaply, otherwise the pharma business would cease to exist. And so they're going to go on being expensive. Which government will pay for them? Will you pay for my amyloid treatment in Israel if it costs $100,000 a year? Or if it costs $10,000 a year, or, you know, and the National Health Service, the same thing, and America. At the moment, the whole drug industry of the world is subsidized by the American payer. Okay? If the Americans weren't paying what they pay for the drugs, there would be no pharma industry at all. It absolutely, 100% depends. They're the only people who pay, you know, over the mark enough to fund the whole business for the rest of the world. So a slightly depressing end note, but on the other hand, it's great that we've got this far. Now, somebody told a joke at the beginning of this session this morning, and I thought I would share one with you if there's time. Have you got time? 2 seconds, quickly, 3 seconds, yeah? You mentioned very kindly my knighthood. So I don't know if those of you who don't know, when you get knighted by the Queen, you have to kneel down in front of her. She taps you on the shoulder with a sword, and then you get up and you're Sir Mark or whatever it is. So there was a famous Jewish philanthropist in London who was knighted, and some years ago. And in those days, when you knelt down and she tapped you, you had to say some sentence in Latin. And this gentleman, who was a billionaire, and he was so good at business, but he was very bad at languages. He wasn't an intellectual, really, and so on. And he had agony trying to remember this sentence. He was learning it for weeks beforehand. And when the moment came, and she tapped him on the shoulder, and he had to say the Latin thing, it had completely gone out of his head. So he said the first thing that came to his mind. He said, "Ma nishtana halayla hazeh." And the Queen turned to Prince Philip and said, "Why is this night different from all other nights?" Questions? I don't know. I can answer questions afterwards.